Okay. Come on, noodle.
Yep, that makes sense.
I clicker. So when you're done with it, the exams go in the black basket. I'll put the black basket up here on the chair. And just throw them in there when you're done. And grades have been posted for a while, but okay, so make sure you check in on iClicker. Make sure you check in on iClicker. Okay, I'm gonna minimize this. We're gonna view full screen. Okay.
And then even like 20 minutes to do it, I still messed up.
My graph, I had it great, and then I noticed that my value for the whole cell and the 65 calorie on the graph was flipped. And so I went to go change it, like how I have that
Yeah,
thing,
yeah.
the y equals
Yeah.
those two numbers, they were flipped, and I went to go change them, and I messed up my graph completely.
Okay, you're gonna have time in lab today.
Okay.
Like there'll be some wait times.
Okay.
So if you want to work on it, you can.
Okay. By any chance have your computer with you, or?
I don't currently.
Okay,
but
because I have on mine, I tried
can-
fixing it on my computer, and that's where I messed it up, and I can't get it back.
Well, I can help you with that.
Okay.
I can help you with that. So just bring your. Do you have your computer?
Yeah, I was gonna
So bring it to
because
lab.
I have after this, I don't have my class that canceled, so I was gonna try and then just because they have computers, so I was gonna try and figure out if I can do it there. I don't know if
It shouldn't matter what computer you're on.
Yeah. So
It shouldn't matter what computer you're on.
because like it let me add the value back. I just couldn't find it. Like it added it.
Just bring it and we'll look at it in lab.
Okay.
We'll look at it in lab. Yeah. Okay, what time is it? I don't even know what time it is.
10:44.
10:44. Oh, okay.
10:45.
10:45, it's time. What is worse than raining cats and dogs? Hailing taxis. I like that. So y'all know we were talking about sugars and stuff. Did y'all hear on the news that there was a guy who actually ingested some L-glucose? Did you hear about this?
Oh.
They all want to know if it's a joke. So he really had no ill effects because he was ambidextrous. It was a joke.
I don't know what I expected, but it wasn't bad.
Wasn't that good though?
Huh?
Isn't that good though? He was ambidextrous. Okay, so on some real science stuff. So we need to talk about stage 2 of glycolysis, right? That's what we're on. We got through stage 1. Stage 1 is our investment phase, right? Stage 2. So let's talk, because that's what we're going to do today. I just gave 2. I just gave 2. It's a Friday. It's a Friday. I'll tell them to begin at the end if you want. All right, stage 2. So in stage 2, what do we call stage 2? Like stage 1 is investment, stage 2 is earnings. Payout, whatever you want to think about it, right? Okay. So what happens in stage 2? What do you start with in stage 2? You start with what kind of molecule? Nope. Glyceraldehyde. Glyceraldehyde, 3 phosphate. You start with 2 glyceraldehyde, 3 phosphates, right? So you have 2 glyceraldehyde, 3 phosphates. You could say per 1 glucose. Should you have started studying glycolysis? Yes. Yes. Okay, the really cool thing about stage 2, why is stage 2 earnings? What are you making? You're making ATP. How many ATP do you make in the earnings stage? 4 ATP per original glucose molecule, right? So how do you generate 4 ATPs? How do you make them? How do you make ATP? Substrate level. Oh, beautiful. Substrate level phosphorylation. So what does that mean? Substrate level phosphorylation. That's this beautiful term. It is. What does it mean? So you have an ADP. That ADP gets converted to ATP. Okay. So ADP is our substrate, and we're phosphorylating it to make ATP. All right. So we generate 4 ATP, but tell me what is the net for glucose for glycolysis? Net is really 2 ATP. Why? Because you used 2 in investment, right? In the initial investment stage, you use 2. Y'all with me so far? Is this good to have a little intro before we jump into slides? Little reminder of what we're doing? Okay, so now, that's great that we're going to generate ATP, but it's not the only thing that we generate. What else do we make? We make 2 NADH. So we make 2 NADH. Anybody know what that is? What it's made by? Which reaction? The oxidation that's catalyzed by glyceraldehyde 3-phosphate dehydrogenase. It's a very long name. So you know what people call it? GAPDH, GAPDH. As if you needed an abbreviation, another abbreviation, right? Glyceraldehyde, P-phosphate dehydrogenase. I just didn't spell out the whole thing. Right? So what's up with these 2 NADH? What you do with them? Where do they go? So they go not to citrate. Citrate also makes NADH. They're going to go to the mitochondria so that we can use them to make ATP, right? Electron transport chain. That's right. Okay. Now, tell me, what is the most important reaction in phase 2 and why? It was on a table and it had some things highlighted in blue that were very negative. The last one, it was the last one. That's reaction 10. It's really, really important. Reaction 10 is pyruvate kinase. And why is this reaction that's catalyzed by pyruvate kinase important? It has a very negative delta G, so it is irreversible. Why would you want the last step in a process to be irreversible? To make sure that the product that you make stays the product. That's really important, right? That it seems simple, but how's the cell going to do that? When you have all of these reactions that can be reversible, you make the last one irreversible. So you ensure, what's our product? Pyruvate kinase, pyruvate, ensure pyruvate is the endpoint of glycolysis. Correct. Correct. You can't go backwards. Once you get to pyruvate, you're stuck in pyruvate. Then you got to do something with the pyruvate. Now, there are multiple fates to pyruvate. 3. And we're going to talk about them. All right. So now let's, can I go up? Are y'all got this? Now let's go do some slides. Is that a good little ease you into it on a Friday? Okay. So when we talk about entering that payout stage, right? We're going to take glyceraldehyde 3-phosphate. And what are we going to do to it? If you look at glyceraldehyde 3-phosphate, what does it already have attached to it? 1 phosphate group, right? And so basically what we're going to do is we're going to attach another phosphate group to the other end of the molecule. So that instead of just being glyceraldehyde 3-phosphate, now we have to specify which carbons possess the phosphate group. So those are carbons 1 and carbon 3. And because there are 2, we call it bis. So it's 1,3-bis phosphoglycerate, right? Now, here's the deal with this. Why is this important? For multiple reasons. We talked about the conversion of NAD+ to NADH and why that was important, right? But the other thing that's important is this molecule is highly unstable. So is it going to hang out for very long? No. So what will happen to this? Again, this is the same substrate, highly unstable. It's the same thing. It's 1,3-bis phosphoglycerate. In order to regain some of that stability, we're going to take off a phosphate group and give that phosphate group to ADP to generate ATP. Oh, awesome. So this is our first substrate level phosphorylation event. Why is it that we are producing 2 ATP? I only see 1 here. Because there were 2 glyceraldehyde 3-phosphates, which means there were 2 1,3-bis phosphoglycerates, right? Which means I make 2 ATP and 2 3-phosphoglycerates, right? Everything is 2. Remember, look at, did everybody have their pathway? Anybody need, if you need another pathway, I have more pathways. But do you see the fork in the pathway? Right? That's showing you that everything that's happening is happening times 2 in that payout phase. It's always nice, like really, if you need any, come and get them, because it's really nice to have this next to you while you're talking about the entire pathway. You can keep the big picture in mind when you're looking at the individual steps. Okay, so what's our next step? Our next step is just a mutase. where we're actually going to take this phosphate group and we're going to tack it on to this carbon right here. That's in the carbon oxygen right here. So all we're doing is rearranging the phosphate. So we go from 3 phosphoglycerate to 2 phosphoglycerate, right? So it's just a simple phosphoryl shift. But we want the whole point of this reaction, it is really important, so that we can make a compound that can be converted into something that we can do another substrate level phosphorylation event, right? So we need to generate another high energy intermediate. So that's what our goal of reaction 8 is, right? Okay. So the next reaction is an enolase. So what is happening? Look, as we go from left to right, what happens as we go from left to right? What are we losing? Look at your substances. What are you losing? Look at that. Yes, water. So we're losing a hydroxyl here and a hydrogen here. And when we lose those 2 things, what do we get? Oh my gosh. 2 carbons participating in our double bond here, right? This is a very high energy molecule. If it's a very high energy molecule, what's so good about it? It's a very high energy molecule that has what? A phosphate group. Yay, we have another phosphate group that's going to be really easy to transfer to another substrate. So now in our very last reaction with pyruvate kinase, we can take that phosphate group off, add it to ADP, and now make ATP. So it's not till the very last reaction that you actually make net ATP. You go through all 9 reactions in glycolysis and you've just broken even. In that very last step, that's where you net ATP. Isn't that kind of crazy? You go through the whole thing and it's not until the end that you actually get a payout. Yeah. So then what do you end with? You end with pyruvate. Why is it really great to end with pyruvate? Okay. So why is pyruvate good? So pyruvate, P-Y-R-U-V-A-T-I, I'm looking at it. So pyruvate is going to be negatively charged. Why is it important for our product to be negatively charged? So if you look up at pyruvate, right, we're negatively charged. Why is that a good thing? Why do we phosphorylate glucose in the first place? In the very beginning of glycolysis. So it won't leave the cell, it's trapped. Why won't phosphorylated glucose leave the cell? Because it's negatively charged. So why is it great that pyruvate is negatively charged? Yeah, it can't escape, it can't get out. We're going to use it in our next step, right? There are multiple different ways that it can go, but we're not going to lose it, right? We can use this, it's negatively charged substrate that can be used to generate more ATP, right? We're not going to lose it. So when we talk about glycolysis, did I put another one? Yeah. And I just want, hold on. I want to go back to the overall picture of glycolysis real quick. Yeah. So when we look at our overview of glycolysis, right, we've done all of our steps, right? We end up with 2 pyruvate molecules. So what's really amazing about glycolysis is that we have actually generated ATP in the absence of ATP synthase. So I know that you studied in high school biology, the electron transport chain, right? And what's at the very end of the electron transport chain? We drive ATP synthesis using ATP synthase. We have made ATP without doing that. That's amazing. So I don't know how you want to put that, but that's something you need to really keep in mind, that the cell makes ATP independently of ATP synthase. So this is no electron transport chain, mitochondria, no oxygen, right? You don't need any of that stuff. That's amazing. So what are our two probably most important steps in stage one? Stage one is called the investment stage, right? Why is it called the investment stage? We invest ATP. So tell me, which two reactions invest ATP? Hexokinase, and you can look, you're looking at your diagram right now, right? And Phosphofrutokinase 1. People abbreviate that PFK1. Okay. So why are these two really important? Not only are they investment, but what else? They're what? Okay, why do they allow for regulation? They're what? They're irreversible. So these, both of these have very high negative delta G values and are therefore irreversible. Right? Okay, so now how about ATP earnings? Where are ATP earnings really important in the earnings phase? Which reactions? Pyruvate kinase is one of them, and phosphoglycerate kinase. So these are two, so 7 and 10. These are our two reactions where we have substrate level phosphorylation events, right? So these two are our substrate level Do they both have large negative delta G values? No, only one does. Which one does? Pyruvate kinase has a large negative delta G value. Phosphoglycerate kinase does not. So do you need to know? Yes.
Why is it called pyruvate kinase?
Because technically the reaction can go in reverse, but it's just very unfavorable. So therefore we call it irreversible, but it is not technically irreversible, because if it was, our experiment wouldn't have happened. But it can, it's just energetically unfavorable, so it doesn't happen in the cell. Yeah. So some things are irreversible because they cannot go in reverse, and some things are irreversible because energetically they are irreversible. Yeah. As if it couldn't get more complicated, right? Okay, so now we need to kind of talk about how glycolysis can be regulated. Now that we know what glycolysis is, the next question is, how do we regulate it? How do we make sure that it happens when it's supposed to happen? So if you go in one direction in this pathway, what we just described, that's glycolysis. So this happens when blood sugar is very what? High blood sugar, we go through glycolysis. You can go in reverse. You can generate glucose from pyruvate, but you have to use a few different enzymes to make that happen, because we know that there are three enzymatic reactions in glycolysis that are energetically irreversible, so they're not going to happen. So you can't just flip a switch and you go the complete opposite way. So at those 3 points where we have energetically unfavorable reactions, we have we technically have 4 new enzymes that come in to replace those 3 reactions. And if we use these other enzymes, now we can go through gluconeogenesis and we can take glycolysis in reverse. So our 3 enzymes that have these really huge negative delta G values, these are our rate limiting reactions. Right? So all of the other parts of the pathway are going to depend on substrate availability, right? If you have a negative delta G value that's really close to 0, what's going to drive you one way or the other? Le Chatelier, right? How much substrate you have. But if you're irreversible, is that going to be true? No. So you have basically bottlenecks at these 3 reactions. And that's because these 3 reactions are irreversible. Okay. All right. So let's talk about the regulation of these bottleneck enzymes, right? So the first one is Phosphofrutokinase 1. This is a great point in glycolysis in order to implement regulation. So what does phosphofrutokinase do? You have to know this, yes. You have fructose 6-phosphate, and it is going to be phosphorylated, and it's going to make fructose 1,6-bisphosphate. So when we do this, right, we're going to use up an ATP molecule, right, because we added that phosphate group right here to the fructose ring. So do you want this step to occur if you haven't eaten in a while? No, why not? What is your energy charge of the cell like if you haven't had something to eat in a while? Your energy charge is really low. What does that translate to in terms of concentrations of ATP, ADP, and AMP? If your energy charge is low, energy charge low, what does that tell you? I have low ATP, high ADP, and AMP. So if we look at how these things, these energy charge molecules are going to affect PFK activity, we can do that. So if we have something where we're going to add ADP, that's the green line, right? So what happens if we inhibit PFK1 and then add back ATP? Well, we're slow, and then we start picking up more activity because ATP is an activator of PFK1. Another activator of PFK1 is fructose 2,6-bisphosphate. And we'll explain why that is later. So you don't necessarily need to know that so much. But ATP is always going to tell you, like, when's the last time you've eaten, right? If your energy charge is high, ATP is going to be high, and this reaction is going to go. If you haven't eaten in a while, your energy charge of your cell is going to be low. You're going to have lots of AMP, lots of ADP, and those things are going to inhibit PFK1 activity, right? So these are inhibitors. And this is a positive regulator. Okay, so let me ask you this. When you go through glycolysis, right, you're going to make pyruvate, right? What happens to pyruvate? We want to send it to what cycle? What's the next cycle? Citrate cycle, CITR. Why do we call it the citrate cycle? Citrate is one of the very first products in the cycle. So how do we get pyruvate to citrate? We're actually going to put pyruvate into the mitochondria and we're going to convert it to acetyl-CoA. So our acetyl-CoA then gets combined with oxaloacetate to go and make citrate. And so if you go in the citrate cycle, eventually the citrate cycle, the final product of the citrate cycle is going to be oxaloacetate. So do you see how this turns into a cycle, right? So if I have a ton of citrate around, do I want glycolysis to continue? No, not really, right? What do I want to happen? I want to go to, instead of using up my glucose in glycolysis, what am I going to go do with my extra sugars? I'm going to go store it, right? What could I store it as? Glycogen. I could also store it as fats, right? There's all kinds of different things that I can do with it, but you have to think about what's going on in the cell and what are my other alternatives, right? Make sense? Okay. All right. So how does your body know when to pull in glucose from the bloodstream, right? Do you want to pull in glucose from your bloodstream when you've only had a little bitty snack, like just a little bit? No, you only want to do this when your blood sugar is very high. So what's the difference between the enzyme that phosphorylates glucose in all other cells and cells that are in either the liver or the pancreas? What's different? One is glucokinase, one is hexokinase. Hexokinase is in everything but the liver and pancreas, right? So what happens in the pancreas? We eventually have insulin signaling, right? So how does it work? Glucose is going to enter the cell just like it does other cells, right? So it goes through a transporter. It goes through a GLU2 transporter. Once it comes in, once the concentration of glucose is high enough, it is going to stimulate activity of glucokinase, right? But this is only in high blood sugar, right? Why is it only in high blood sugar? Because glucokinase has a lower affinity for glucose. So you could say than hexokinase. Hexokinase doesn't care. Hexokinase binds really tightly to any little bit that's inside the cell, right? Glucokinase waits until the concentration gets high enough. And this is really important because that's the initial event that is going to trigger insulin release. So when we have enough glucose, glucokinase begins. The rest of glycolysis is exactly the same. It's no different than any other cell. The only step that's different is glucokinase. So once we go through glycolysis, we make pyruvate. We're going to go through the citrate cycle. We're going to go through the electron transport chain, and we are going to make tons of ATP. Once that ATP is made, it's actually going to inhibit a potassium ion channel. You remember how we talked about different ions being on one side of the membrane than the other side of the membrane, and you can get a potential difference? And this is how our nerve cells work in communicating, right? So it's very similar. If you inhibit this potassium channel, you are actually going to get a depolarization of the membrane. And once that depolarization happens, you're going to actually open a different channel. You're going to open a calcium channel. When you open that calcium channel, now the calcium comes in and it activates these vesicles. When these vesicles are activated, they go and merge with the membrane. Well, guess what's inside the vesicles? Insulin. This is why you only release insulin when your blood sugar gets high enough, right? So how important is the difference between glucokinase and hexokinase? Super important, right? Which are, because glucokinase has lower affinity for glucose than hexokinase. Yep. Okay. So we've talked about glycolysis in terms of just glucose, but is that the only sugar that we ingest? No, we ingest all kinds of different sugars. Maltose is an example of that's a plant starch that we ingest. And maltose is a combination of what two sugar monomers? Glucose and glucose. It's two glucose. So if you have the enzyme that will break down maltose, what are you going to make? Glucose. So you can directly enter glycolysis. If you drink milk, inside of that milk, you have lactose, right? What is lactose a combination of? Glucose. So if you cleave it, you can have glucose directly enter glycolysis. What else can you make? You can make galactose. Now, can galactose enter glycolysis? No, it's a problem. We need to convert galactose into a substrate that can be an intermediate in the glycolytic pathway. So we're going to convert galactose by phosphorylating it and then converting it to glucose. So we start out with galactose 1-phosphate and we convert it to glucose 1-phosphate. Glucose 1-phosphate can then be converted to glucose 6-phosphate. Now that's one of our intermediates. We're good, right? Sucrose is another example. Sucrose is table sugar, right? What is table sugar? Fructose and glucose. So the glucose can go directly into glycolysis, but our fructose here has to have something done to it, right? And it kind of depends on where you are exactly what's going to happen. But you can just phosphorylate it and you can go from fructose to fructose 6-phosphate. Now I can enter glycolysis, right? So what happens when you're going through glycolysis and we have extra, right? We can go in and we can actually make fats. We can use the carbon backbone in order to make fats. When you talk about triglycerides, what are triglycerides made up of? They're made up of a glycerol molecule and 3 fatty acids, right? So these pathways can go forward and reverse. So if you go on a low carb diet, right, what does your body start to do? We still want to use glycolysis, but we don't have any sugars because we went on a low carb diet. So we're going to take those triglycerides, we're going to break off the fatty acids. The fatty acids we can break down through oxidation and make acetyl-CoA, which can then go in and enter what? Citrate. And then we also make a glycerol molecule. We can convert that glycerol to glycerol 3-phosphate, and then we can convert that to dihydroxyacetone, and we can actually enter glycolysis. So there are lots of different interactions between glycolysis, different substrates, depending on what the organism has eaten and hasn't eaten, right? I don't want you to think about it as just a simple linear pathway. I want you to understand that different things can feed into it at different points. Does that make sense? Yeah? Okay. And you don't have to worry about galactose metabolism. I'm not going to test you on that. I'm not going to test you on that. All right. So when we talk about what else can these products and these intermediates be used for, that's a really great question, right? So we know that we can take glycolysis, we could go and we can make pyruvate. But there are other things that you can make, right? So what we've talked about already was I can take glucose, I can phosphorylate it, I can trap it in the cell, and then I can make glycogen, right? If I have a lot of excess glucose and my ATP levels are really high in the cell and I don't need any more ATP, I can go and store it. I can then do the same thing by taking the glucose, converting it to dihydroxyacetone, go to glycerol 3-phosphate, and I can go make me some triglycerides, right? This is the body preparing for winter, right? Winter is coming. I'm going to make some triglycerides. The other thing that we can do with glucose is, you remember when we talk about our nucleotides, what is one of the components of a nucleotide? A sugar. A sugar. So do you need sugars to make DNA? RNA? Absolutely. So at the same time, glucose can be shunted off to what's called the pentose phosphate pathway. And when it goes through the pentose phosphate pathway, some of those intermediates can be converted into nucleotides, which is really, really important. You can also make all kinds of different amino acids with different substrates, depending on where you are in the pathway. There are some here, there are some here. You remember when we talked about hemoglobin, and we talked about one of the inhibitors of oxygen transport was 2,3-bis phosphoglycerate, right? We can convert some of these substrates into 2,3-bis phosphoglycerate, so we can regulate oxygen transport. The other thing that sugars are used for, we didn't talk about it so much in this class, right? But you know about blood types, right? So our blood types are really just different carbohydrate molecules that are sticking out on our red blood cells. And lots of cellular communication happens with proteins that have carbohydrate groups or with lipids that have carbohydrate groups. So if it's a protein with a carbohydrate, we call it a glycoprotein. If it's a lipid, we call it a glycolipid. But these are super, super important in all kinds of different cell signaling that occurs from one cell to another. So there are lots of different ways. This is very, very much a network of things that are happening inside of the cell. It's just a fraction of them, right? Okay. So we've made pyruvate, right? We're so happy we've made pyruvate. What is going to happen to that pyruvate? There are 3 different things that could happen to the pyruvate, and it depends on 2 things. What kind of an organism you are in, and whether oxygen is present or not. If you have aerobic conditions, aerobic means what? Oxygen is present, right? Then we can, and where do we go? Right? We were in the cytosol, but in order for oxygen to be present, where do we have to move? We have to go to the mitochondria, right? So we ship it to the mitochondria, we convert it to acetyl-CoA, we go through the citrate cycle, we go through the electron transport chain, and we make ATP, right? That's one fate. But if you don't have oxygen, you can either make lactate or you can make ethanol. So what is something that we like to eat that is due to the production of lactate? Milk, cheese, yogurt, right? All those things that we like to eat. And then ethanol production, if you enjoy a nice beer, you have anaerobic conditions and pyruvate to thank for that. So let's look at kind of how that works. So it's really, really, really important what happens to pyruvate. The number one thing that we have to worry about is, can we regenerate NAD+? So remember when we were going through glycolysis and we used 1 molecule of NAD+ and we made NADH? Remember that? And we said, okay, we're going to make that NADH. And where's that NADH going to go? eventually it's going to get to the electron transport chain, and we're going to use it in order to use our proton pump in order to run ATP synthesis, right? So if that happens, you have to have a really elaborate shuttle system. So if pyruvate goes to the mitochondria, you must have a shuttle system to regenerate NAD+. And we won't talk about that yet. We won't talk about what that shuttle system is. But because you're transporting it from one subcellular location to the other, right, you have to have some kind of very complex system to get NAD+ back. And we'll get to what that is later. If pyruvate goes to mitochondria, you have a shuttle system to regenerate NAD+. So what are the other two possibilities? Let's say that we don't go through the mitochondria. The big deal is that we can regenerate NAD+. So there are two things that can happen. Let's say you have a muscle, right? And you've been exercising a lot very quickly. What happens? You deplete what? You deplete oxygen. So you have no more oxygen, but you still need energy. If you don't regenerate NAD+, you're not going to make ATP, your muscles are going to fatigue, and you are going to have some serious problems. So what happens instead? You make lactate. So using lactate dehydrogenase, pyruvate is converted to lactate. In that process, NADH is converted back, see here, converted back to NAD+. Now we're ready to go through glycolysis again. So you can have people who have mutations in their lactate dehydrogenase genes. And when they start doing really strenuous activities, their muscles fatigue very quickly because they have no backup system to regenerate the NAD+. And so they go in these extreme conditions where they don't have any ATP. And it's actually very, very damaging to the cells. And they have a lot of different symptoms and things. So lactate dehydrogenase is very important. What protein did we purify in lab? Lactate dehydrogenase, right? Very important, very important. So this is the enzyme that we've been working on for I don't know how long in lab, right? Yeah. So this depends on, right? You could do this in your muscle. There are some bacteria that use this, and that's what we use to produce yogurt and things like that. But the other thing is if you have Saccharomyces Cerevisiae, right? What these organisms do is they take pyruvate and they convert it through a fermentation process into carbon dioxide and ethanol, right? And so this is how we get, that's what they call it, brewer's yeast, right? So I worked in a lab where we used Saccharomyces Cerevisiae, and it smelled amazing in our lab. It either smelled like beer or bread. It just depended on what strain people were growing at the time, you know? So it was really fun. And you could tell, it was really funny. We were always worried about bacterial contaminations into our cultures. And you can actually smell the difference. Like you can smell a yeast culture that has been contaminated by bacteria because you have different scents. Very interesting. But the whole point is that whether we go through fermentation, whether we go through lactate dehydrogenase, or we go through aerobic metabolism within the mitochondria, that we are regenerating NAD+. Because if you don't regenerate NAD+, you're going to deplete your cell's ability to go through glycolysis anymore, which is really dangerous, right? So NAD+ regeneration, this is what's really important. So this is how we maintain our flux. And if you look at what reaction in glycolysis requires NAD+, which reaction is it? gap DH. So when we convert from glyceraldehyde 3-phosphate to 1,3-bis phosphoglycerate, this is where we use up one molecule of NAD plus and we make NADH. And before we just said, oh, NADH is great, we can take it to the mitochondria and we can use it right in the electron transport chain, which is true. But in the mitochondria, we have a shuttle system that allows us to regenerate that NAD plus, and then it can come back and go again, right? But it's technically regenerated in the cytoplasm, and then you have this shuttle system that will take a new reduced molecule to go and participate in the electron transport chain. So this is lactate dehydrogenase deficiency. This is where you have a patient who has that mutation in the enzyme, right? So either maybe it's a mutation in the enzyme, maybe it's an expression problem that they aren't making enough of them, whatever it is, the enzyme can't work. So if your enzyme doesn't work, then you're not converting NADH to NAD plus, right? And so we don't have that regeneration of this molecule anymore. And when we don't have regeneration of this molecule, we no longer go through glycolysis. And if we no longer go through glycolysis, we cannot have anaerobic production of ATP. So you'll see these people can't exercise. They can't, because they're actually do more damage to themselves than anything else. So it's a really sad disease, right? And then the very last one is alcoholic fermentation, right? So we're not going to go through all the steps, but basically what you're going to do is you're going to use those products of glycolysis, right? And when you undergo fermentation, the sugars, right, that are made there are fuel for our yeast, and our yeast are going to go in and make delicious, wonderful alcohol, right? Okay, so I want to talk about, so I'm not going to test you on how you make beer. If you want to know somebody who makes beer, right, a biochemist that makes beer, Dr. Smith makes beer. He makes some very, very delicious beer. It's very good. It's very good. Okay, so I want to talk about kind of glycolysis in general. How much time do we have? Oh, a few minutes. 6 minutes. We can do 6 minutes, right? Okay. So let's try to summarize and talk about a few big points, right? Okay, why is glycolysis great? What is so great about glycolysis? Because, why is it great? Right? It is great. What do you make? You make ATP, but not just any old ATP. You make ATP independent of what? Independent of two things, right? Oxygen and ATP synthase. What's the other thing? What else do you get out of glycolysis? You get, so glycolysis, we'll say makes, what do you make? 2-pyruvate. What else? 2, not 2. How many NADH? 2 per glucose molecule. How many ATP? Net 2 ATP. So you've made some energy carrying molecules. You've made ATP, you've made NADH, but I want to talk about this pyruvate. This is what I really want to talk about. You make pyruvate. What happens to that pyruvate in typical cells? I'm not talking about fermentation, right? But I'm talking about in us. We can use that. We now have a product that can be used in subsequent energy production pathways. And what's so great about pyruvate? It's stuck, right? It can't get out. Why is it stuck? And cannot and can't leave because it is negative. And that last step was irreversible, so you can't go backwards. Okay, so without looking, don't look at your pathway. Can you tell me what are the investment and payout reactions? Just take a second, write it down. Investment and payout reactions for glycolysis. Take a second and like jot them down. So my short answer for this is you should have done 1, 3, 7, and 10, right? You can go back and look and make sure you get those right, but go double check and make sure you know which ones are investment, which ones are payout. What are the substrates? What are the enzymes, right? Okay, now, which of the reactions are irreversible? What do we say? 1, 3, and 10. What do you notice that's really similar? 10, right? Like if you know what the investment and payouts are, you also know it's irreversible. So it'll help you with that, right? So when you're studying, don't just study, okay, this is the reaction, but why is it important, right? Where are we going to regulate glycolysis? Why is regulation of glycolysis important, right? All that kind of stuff. Yeah? Okay, that's it. So 1 minute. Citrate cycle to next week. Awesome. Yes. And there's one problem on the homework that I'm contacting them about that I don't think it's worded very well. So you'll see that eventually, that everybody's going to get credit for that. Y'all have a great weekend.
I don't know if we can do anything about this now, but I was looking at it. I circled D. Somehow, I don't know why. I accidentally put C. I didn't have time to go back and check because I ran out of time. But I mean, you can't do anything about it. I'm not worried about it. It's not telling me. Well, the bubble should have put C. Yeah. So, yeah. I figured, but.
Yes. Sure.
It has the opposite for what inhibits PKPFK1, and it has ATP as an inhibitor.
So if your energy charge is low, right, that means you haven't eaten in a while. So what do you want to do? You want to metabolize the sugars that you have, right? So you want to go through glycolysis. So you want to activate it.
Okay.
If you have eaten recently, you've been through glycolysis, you've made tons of ATP, you're in the citrate cycle, you've made a ton of citrate. Do you want to go through glycolysis anymore? No, you want to prepare for winter, right? Winter is coming. So you're going to inhibit the glycolytic enzyme PFK1.
Okay. So it kind of just depends on if you have high sugar concentration versus low sugar concentration.
Well, right, which is gonna tell you, if you have a high sugar, you're gonna go through glycolysis, you're gonna make ATP, then ATP is a product gonna come up and inhibit it. It's like a feedback. It's the same thing with citrate. Citrate's just lower down on the cycle, right? Because now you're in the citrate cycle, but it's still a product of glycolysis that's gonna come up and inhibit it.
Okay.
So that you're not investing ATP when you've made tons of ATP, right? You can go off and make glycogen, you can go off and make fats, you can do all that kind of stuff.
Okay. Thank you.
Yeah.
So my first question is, I think Shoot was just asking you this. So I thought that you said in class that you said if you didn't eat, then you wouldn't make any more. But I thought if you didn't eat, you would want to
If
make
you're down
more.
ATP, you want to activate to make more.
Yeah.
Yeah.
I
What did I
must
say?
have heard. I think you said.
Did
Yeah,
I,
you
did I write it wrong?
I think
Sometimes
so.
I'm talking and I'm not looking at
I
it
know,
right,
it's-
wrong.
Yeah.
Okay, yes.
So it should be the opposite?
Swap it.
So this is a negative regulator.
This one is a negative, because if you
Okay.
think about it, right, you're going through glycolysis. You have glucose, you're gonna use that glucose, you're gonna make pyruvate. That pyruvate is gonna end up going to acetyl-CoA, going to your citrate cycle, right? And in the citrate cycle, one of the first substrates is citrate that's made, right? So glucose is making ATP. Citrate is making ATP, basically. I mean, you gotta go through electron transport chain. But these are two products. Y'all can come in, it's okay. So this is feedback inhibition, right? So I want these products, these are all products, to come back and inhibit the pathway. So these are going to inhibit PFK1, right? So the other part is true. You haven't eaten a lot, right? So do you have a whole lot of ATP? No, like you haven't eaten. You're hungry. You don't have citrate. What do you have? You have ADP, AMP. Those are going to be positive regulators of PFK1. I think I must have written it on the screen in reverse.
Okay, that makes sense. So positive regulator, and then ATP is an inhibitor.
Right, because you haven't eaten. You're hungry. So do you want this glucose to be used for, because glucose can be converted to pyruvate, and pyruvate can go to fatty acid synthesis, it can go to glycogen storage, or you can go and make ATP, right? So if you've got lots of ADP, lots of AMP, you want to continue to go through citrate, right? You want to continue to promote this. But if you have tons of ATP, what do you want to do? Go store the junk that you got extra of, right? Winter's coming. Prepare. Yeah?
That makes sense. Okay, I have two more questions.
Okay.
So let's see. Let me pull up my line report. So whenever I printed out my Excel, it came out like teeny tiny. I don't know why.
That's okay, but you're gonna upload it to
I
Google.
did. So like it's, oh, I'm sorry. I'm
Oh,
losing
wow,
sound.
it's really teeny.
Yeah, it's
That's
because my
okay.
table came out
That's
big.
okay.
Okay.
That's okay. So what you can always do in Excel is you can highlight the parts that you want, and you can do print to, you good? You have a question?
No, I'm good. I survived for another test.
Good. So you can highlight and you can say fit to 1 page width or whatever you want.
Mhm.
But that's fine. That's fine.
Okay.
If you stable it like that, that's fine.
Okay, thank
Is
you.
this you?
Yeah, that felt in my binder. Also, if I haven't, I don't know what I'm doing wrong with it. Like, if I have another clicker thing, should I just email you about it?
Just that it marked you absent when you were
Yeah.
present. So every time you leave out the door, swipe
I need to
clicker up.
close it and open it again.
Yeah, just get rid of it.
Because sometimes I get error messages. I don't know. So
That's-
maybe I just need to keep.
I have a question about the
Sure.
test, and, okay, so number one,
Yeah, this is the one that
I'm having a lot of trouble with.
number one's wrong. Number one's wrong.
I've done,
So-
I did multiple ones, and I'm like.
It's wrong. It's wrong. So I emailed them about it, but I haven't heard back from them yet. But this is the one everybody's gonna get credit for because it's wrong. So once I have it to where everybody's gonna get credit, I'm gonna send out an email that explains it.
Okay.
But you're right. So
I didn't know, okay.
because they were
because it
talking
says-
about this too.
Exchange of compounds, it does not, and I was wondering if it were a correct one, would you say that animals requiring oxygen for phosphorylation, is that an exchange that's going on? Or is that technically something you should check? Well, it's not technically an exchange going on between, just having this statement here, that's not necessarily saying it's an exchange, right?
But it does.
Because I can
There
see-
is an exchange because you require oxygen for phosphorylation of ATP through ATP synthase, not through glycolysis, but through ATP synthase, right?
This is a correct statement, right?
It is a correct statement.
Okay, but is this an exchange between animals and plants?
Yes, because when you make that ATP, right, one of the byproducts of making that ATP is CO2. They just don't say it in this statement.
That's what I was wondering. I didn't know. if
They
that was
don't
technically
say
an
it,
exchange.
but it's true. It's true. You require oxygen for
Right, that's-
phosphorylation of
Right, I knew that
ATP.
was right. I
And
was like,
then-
is there an exchange between plants and animals in this? Because I feel like it's such a small exchange, like us breathing, exhaling CO2, and then carbon fixation through the CO2.
Correct.
It's so tiny that it was almost like, I don't know. if
It
this is
does.
actually-
It does.
Okay.
You gotta
I
think
just
about
didn't
all
know.
the animals
I
on the
was kind of
planet.
second
It's
guessing
not just,
everything. at this
not
point,
just
but
you and I know. No, I really don't like how they did that.
I really don't like their program, to be honest with you. Because it's just, there's a lot of things that they make mistakes in, and in the book as well.
Well, the book is because it's the new edition.
I know it's a big.
They're
the money grab.
ironing out the kinks.
And then, okay,
And to me, it should have been ironed out before, but
I haven't tried this yet
Okay.
on the space mission with. the-
yeah,
Okay, wouldn't it be, they produce too much ethanol? and they don't?
open the closed containers.
Because that is like something that does happen when you accidentally,
Yeah,
apparently online I was looking at that
So-
one's wrong, and I'm wondering, but I feel like that one should be.
does it ask you which one's wrong or which one's right?
What do you think would
Elimination.
happen if you opened up the two? and you
With
looked at
ice,
something?
the researchers in time when they first tested out the experiment in lab by growing yeast under simulated microgravity for a few
I
hours.
feel like they left it, but like maybe it blew up. I think it says the container blows up. Maybe,
Well, so what are you producing when you undergo fermentation?
maybe all CO2.
You're producing
So, like,
CO2.
maybe it's possible that it does.
So you are producing CO2. Open to discover stimulated and cause the cells to reduce their rate of glucose. Should no significant growth. No, they're still gonna grow.
So it's thinking they either died.
None of the cells have grown due to lack of oxygen. But do you need oxygen? If it's Saccharomyces Cerevisiae, you don't need oxygen. You're gonna produce CO2, absolutely. Significant amount of ethanol had been produced under these conditions, and the yeast had begun to die as a result. So they are gonna produce ethanol.
They do put in, after a predetermined growth period, fixative auto injected into the chamber to stop growth to protect. So they know that they're gonna slow
Mhm.
growth, which I feel like B would be, that would be understandable. They would see that. So,
Yeah, they.
but it wasn't due to lack of oxygen.
It wasn't due to lack of oxygen,
Okay.
right?
It'd be due to the fixative. though.
Right, but they're not adding, y'all can come in. I'm sorry, y'all can come in. Come on. Y'all can come sit down. It's fine. Yeah, go get your bag. Go get your bag. We're gonna walk so that we're out of there. Right. Oh, I'm still, look at this. I'm still on Zoom.